All right, so this is the chapter on tissues and the integumentary system. Right. All right, so the tissues. So we looked at, in chapter one, all your terminology, directional terms, things that help you work on vocabulary to discuss anatomy and physiology. Then we talk about chemistry. And inorganic and organic chemistry, types of reactions, types of bonds, functions of these things. Then we move on to cells. And then cells come together to form tissues. Tissues come together to form, systems. And systems come together to form an organism. So tissues is how we're going to organize all these tissues or talk about the different types of tissues. And then we go on to the integumentary system, which is our first system, of the body that we're actually going to be covering. So all tissues of the body, and it's sort of a generic term, but when different cells come together with a common function or common goal, it's called a tissue. And we have four basic types. Everything that you look at for the rest of your life, when it comes to tissues, will fall into one of these four categories. And our little acronym is SEMEN. Connective, muscle, epithelius, and nervous tissues, or epithelium, epithelial, and nervous tissues. And so everything we look at falls into one of these four major categories. And when we look at some examples of these things, it becomes, fairly easy. Muscle and nervous tissue, there's not a lot of, subcategories to it. Connective tissue has the widest variability within it, and epithelial tissues are found in certain places. And then there's a way that they're all named and what these terms mean. So whenever you look at the tissues, often the word connective or muscle or epithelial or epithelium or nervous, is in the title of the tissue. For instance, types of connective tissue are like adipose connective tissue or dense regular connective tissue. When it comes to epithelial tissues, it might be simple squamous epithelial tissue or epithelium. So often the category of tissue is sort of in its last name. So it's giving you a hint. It's like, okay, what kind of function might it be? What kind of characteristics, might I be looking for, in this type of tissue? So connective tissue binds organs together, supports and protects the body, stores energy. It has multiple functions. It kind of depends on what kind of connective tissue it is. And that's why I was saying it has, a lot of variety. But connective tissues will never be found like on the surface of a body, or they'll never contract and relax and cause movement like muscle does. Muscle cells are designed to shorten and lengthen and shorten and lengthen. We have three different types of muscle cells, but that's all they do. And in doing so, they generate heat. When they contract, depending on where they're located, like smooth muscles, they might propel substance within, a tube or a hose, so to speak, along its pathway, kind of squeeze behind it like you're squeezing toothpaste out of a container. Epithelial tissues line hollow organs, cover surfaces such as the body, and form glands. They are characteristically avascular, which means that they don't have a direct blood supply. They often get their nutrients through diffusion. They're generally very highly innervated, and they have a high mitotic rate or a rate of regeneration. And we looked at mitosis, interphase, and then the mitosis phases, prophase, metaphase, anaphase, telophase, in the last lecture. So epithelial tissues, especially an example is your skin. The surface of your skin is composed of what's called stratified squamous epithelial tissue. And as we all know, that your surface of your skin will go through periods, sometimes more, sometimes less, of getting dry and kind of exfoliating, and flaking off the surface. Well, those cells originate from, the the inferior aspect of the epidermis will look at that, called the stratum basale. And from this one point, these cells go through high mitotic divisions. They replicate over and over and over again very rapidly to push the cells from the bottom upward toward the surface and keep replacing that tissue that kind of gets dry and exfoliates from the surface of your body. That's why we often use moisturizers on the surface of our body to keep it nice and moist. So epithelial tissues generally demonstrate high mitotic rates. And then nervous tissue, like the nervous system, is a form of communication, within your body using electrical impulses mostly. But we also have some other cells that don't necessarily conduct an impulse or an electrical message of some kind, but they help those neurons do their job in both the peripheral and central nervous systems. So it's used for internal communication, detects changes in the environment, help generate responses to maintain homeostasis. It's kind of a broad category, but there are only two major subdivisions of the nervous tissue. So we'll take each now. So epithelium linings, or covers, epithelial tissue or epithelium are linings and covers. Now, when it comes to the naming of epithelial tissues, we often have a couple terms that we use to describe certain characteristics of this tissue. The first term is generally the number of layers of cells that you're looking at. The layers of the cells. I'm going to give you an example in a minute. So simple means it's a single layer of cells. That's all it means. Simple, like simple squamous, refers to one single layer of, we're going to look at a squamous cell given its shape. Stratified means two or more layers that have a protective function. That's what we find in the skin. Pseudo-stratified means it appears to be stratified, but it's only one layer. And transitional means it appears to be stratified, multiple layers, but when stretched, it's usually about a 3-layer thick, or 3 layers of cells thick. So I'll give you an example that we especially look at a lot of transitional epithelium in the urinary system, where we have the pathway of urine once it's formed from, what's called the renal pelvis to the ureters to the bladder, and then parts of the urethra, as that's the tube that transports urine from the bladder to outside the body. And those areas of the body are where, we have essentially a collapsed balloon that can distend and stretch when urine is passing through it, but will collapse back down when there's no fluid passing through it. And that's so air and things doesn't accumulate in there. So that tissue needs to be able to stretch when necessary and not rupture and collapse back down again. So there's certain areas that we often find, like pseudostratified ciliated columnar epithelial tissue, is along primarily the type of tissue or epithelium that lines the respiratory system. Stratified squamous is going to be on the surface of your body. It's going to be parts of your mouth and your throat where food passes because it's very rough. And stratified means it's multiple, many layers of cells that you're looking at. And so, where you sometimes have abrasion to that surface, like on the top of your skin, on your body, on the surface of your body. And as you chew and swallow food, that's rather abrasive. You're going to have that type of tissue there. Okay, so let's move on to the second term. The second term refers to the cell shape. It's either squamous, cuboidal, or columnar. Squamous cells are kind of like a fried egg. They're flat with a nucleus in the center that might hump up a little bit, but they're rather flat, and the edges of it are not, you don't have a lot of long flat edges. They kind of are hexagonally shaped, kind of undulating like a fried egg, but it's not going to be like a cuboidal or a columnar. Cuboidal and columnar cells. Cuboidal cells, the best analogy is a nice square ice cube that might have more shape to it, but you have multiple kind of flat surfaces to it. And, it's about as tall as it is wide, like an ice cube, a square ice cube. Columnar cells are more like a domino, where they're taller than they are wide. And so you find those in certain regions. So the first term will generally refer to the number of layers of cells you're looking at. The second term will describe the shape of the cell you're looking at. And then it's usually followed by epithelium or epithelial tissue. Okay, so here we have on the top, we have a layer that would be called simple, or if we're describing an epithelial tissue, we said it was simple something, let's say simple squamous. This would be an example of simple, one single layer of cells up at the top. The apical surface is the surface of the cell that is exposed to either the surface of your body, like air on one side, or that might be the layer that it lines the lumen of a tube, let's say your respiratory tube, your trachea, or along the pathway of your digestive system where food will pass through, or the first layer, of your heart, that's intimate with the blood that's passing through it, or a blood vessel, the first layer of tissue that would be in contact with the blood that's passing through that tube or hose. Right? So the apical surface is sort of like the head, so to speak. The basement or basal surface is the surface of the cell that will then connect to other tissues, because otherwise it'll just pull off. So below the epithelium, is shown as some kind of connective tissue layer that they will kind of affix to and anchor themselves to. Right, then we have stratified, and the picture below simple represents stratified cells. Right, so multiple layers. In the epidermis, you'll see way more than that. It's really, really thick layers, and that has multiple functions associated with it, like preventing bacteria and viruses, from getting into your body. Right, then we have classification based on the number of cells. Oh, that's above that. Oh, yeah, below that, the classification based on the cell shape. So we have squamous, and if you look at it from the side, it kind of looks like a fried egg. Cuboidal is like a cube, and columnar is like a column or maybe a domino. But you can see here between the squamous and the cuboidal cells, they form those nice tight junctions between adjacent cuboidal cells or columnar cells. Squamous cells, not so much, because the height of it is very squat, it's not very tall, so it doesn't form good tight junctions at all between adjacent squamous cells. They'll often have little gaps in between the two of them, and thus that allows a certain function as well. Okay, so here we have some pictures, histological slides of different tissues. Here in the upper right hand, left hand, I can't tell my left from my right, sorry, up in the upper left hand at about 11 o'clock here, we're looking down on a layer of, from superior to inferior, looking down on some squamous cells here. So you can see that I have that kind of a funky shape to it. From there to the right, to the upper right corner, that is an example, that is a slide or histological slide from the skin. From the dark purple all the way, that large dark purple region, that has an undulating, border to it, or basement to it that undulates like a wave, right? That is all epidermis, actually. And below that, in the lighter, paler staining, lavender, pinkish, with the dark purple spots in it, that is the beginning of your dermis, so the second layer of your skin. But this upper, darker purple layer, that is stratified squamous epithelium. That's what we find in the upper layer of your skin. Composed of multiple layers of squamous cells. Okay, and then at about 3 o'clock, here we have some columnar epithelium, simple columnar epithelial tissue, it's called. You can see at the bottom, you can see the nuclei all in a row, all right, the darker purple. So immediately, or just about immediately below that, is the bottom or the basal surface of that one cell. And it extends quite extensively. It's a rather tall columnar cell, and it faces the material in the lumen there that's kind of a pinkier color. Down below at about 6 o'clock, that would be a great example of transitional epithelium, where you can see all these different cells and it looks like they're multiple layers, but actually when it gets stretched out, it's only about 3 layers. Back to that simple columnar at 3 o'clock there, one way you can tell that that's a single layer is that the nuclei are all rather in a row. They're pretty much in line with one another. You don't have some that are popping way up to the top and down to the bottom and up, bottom, top, and they're squishing all in there. They actually, once, these columnar cells have enough room where all their nuclei can reside down at the more basement membrane, or basal layer of that cell. And so you can see pretty much a rather straight line of nuclei. That is a big key, observation or characteristic or visual cue, that you're looking at simple columnar epithelium. But the tissue at 6 o'clock, they all have the same basement membrane. It's kind of the dark pink line on the bottom. And you see how the nuclei kind of bob up and down and up and down and up and down, where they look like they are multiple layers, but they're not. They're just so squished in there when the tissue is relaxed, or this vessel or ureter, is relaxed, is that they all have to crowd on top of each other. So there's barely enough room. So the nuclei pop up and down and up and down and up and down, so everybody can fit. But if we pass fluid through, or air or something through that tube system, and it stretched and it dilated, then you'd see it stretch out to where you would see about 3 layers of cells. That's transitional epithelium. And then over here at 9 o'clock, beautiful picture. This is from the kidneys, right? These little tubes, you see these little hoses on end? That is a single, each hose that you're looking at, right, with a lumen in the center. That is one single layer of cuboidal epithelium. Right? So that is simple cuboidal epithelium. Each one of those little squarish cells, they kind of look like an ice cube with a cherry frozen in the middle. That's its nucleus. Right? That is a single layer of cuboidal cells. And single cuboidal epithelial tissue, frequently, like 99% of the time, is the type of tissue that forms ducts. And ducts transport products from a gland to maybe the surface of the body, or a gland primarily to the surface of the body, or to the interior, of another organ, let's say from the pancreas along the pancreatic duct to the small intestine, let's say. And these tubes, or these ducts, are one single layer of cuboidal cells like that. They're not multiple layers, they're just a single layer. And thus we can fit a lot of these ducts or tubes or, tunnels, into an organ. Many of them can fit in there. And they're right next to each other. So some material can be exchanged from one cuboidal cell to another. Right, so those are examples of some shapes and some different layers. All right, so glands. Glands are 99% made up of that simple cuboidal epithelial tissue that we just looked at. Types of glands that we're going to look at, especially in this next chapter, are sudoriferous and sebaceous glands. Sudoriferous means a sweat gland, and a sebaceous gland is an oil gland. We have thyroid glands. We have, gosh, all kinds of glands. We have thyroid, parathyroid, thymus glands. We have the pituitary gland, anterior and posterior. We have the adrenal gland, composed of a cortex and a medulla. We have glands all over the body, but some of these that have these common functions like sweat and oil are going to be found, all over on the surface of your body. And then we have some other kind of specialized glands like ceruminous glands. They produce the earwax that you have in your ear, in your external auditory meatus. But they are almost every single mucus glands that we see. Oh, mucus glands line the digestive system and the respiratory systems, composed of simple cuboidal epithelial tissue. All right, connective tissues. Connective tissues are generally, composed of matrix and then the cells found in the matrix. The matrix is the material that fills the space between cells, and it can contain what's called ground substance, which are molecules that support the cells and provides exchange medium between the blood and cells. And then we have different fibers. We have 3 basic types of fibers. We have collagen, elastin, and reticular. And, given that they're different fibers, they have different functions, so to speak. Collagen and elastin fibers, it's not uncommon to find them mixed, between them, having both of those, especially, where we're looking at tissues that need to stretch in multiple directions, like the connective tissue that might surround the heart, or around a blood vessel. Any of these major organs that have to stretch and distend and more than one plane, like blowing up a balloon. They'll often have a mix of those two types of fibers. And reticular fiber generally, that are the narrowest and the darkest staining, are generally found in the spleen and the lymph nodes, because they are part of what's called the reticuloendothelial system, big fancy word, but of these organs, that require the reticular fibers, and they are going to be the narrowest and the darkest staining. Collagen fibers are the most abundant. They're very highly tensile, so that therefore they resist stress. They resist over compression and squashing. They're rather wide diameter, and they have a rather translucent appearance compared to elastin. So elastin or elastic fibers are collagen fibers, and they start taking up a deeper stain, a darker stain to them. They can be like an elastic band, stretched and then recoil and bounce right back again. Well, not completely back again, but they can be stretched and then they recoil, let's put it that way. Most of the time they can bounce right back again. And then we have the reticular fibers that are the narrowest of them and take up the most stain. So collagen fibers are kind of little chubbers there. They're a little translucent. And then they're, a little skinnier are your elastin fibers that take up kind of a dark purple stain. And the reticular fibers that are the narrowest, kind of like a strand of hair, and it takes up the darkest stain of all. The cells, each class of connective tissue has its own cell type in an immature or mature form. In bone, we have three types of cells. We have osteoblasts, osteoclasts, and osteocytes. So osteo means bone, and blast, site, clast, in Latin, they're going to mean certain things, and therefore describes and imparts a certain function to that cell. But we'll look at more the specific functions of cells like that when we get to each tissue. All right, connective tissues, they can also kind of be categorized as either loose or dense, and then either regular or irregular. So loose connective tissue, so loose. Cells and fibers are suspended in a lot of ground substance. So there'll be fibers and there'll be some cells in there, but they have a tendency to have a lot of matrix and fluid around them. So my analogy, which is usually involving food, is like a wonton soup, maybe, where you have a lot of broth, and then you have a few wontons thrown in there. It's a lot of broth, but it has some, material that it's suspending in there. Dense connective tissue, is mostly fibers and cells tightly packed together. So we might have a lot of collagen fibers and cells with very little ground substance. Right? So an example of that might be, let me think of what everybody might think is maybe a stew. If you cook, because everything's about food to me, when you make a stew, or maybe chicken soup, or beef stew, you often have either chicken or beef in there, of course, but you'll have a lot of vegetables in there as well, and maybe some rice or some spaghetti thrown in there or something. But it's generally very hearty because it's very thick, and rather dense. So you have a lot of vegetables and meat and maybe pasta in there, and it's in broth, but not nearly as much broth as maybe like a wonton soup. Right? So the bath it's sitting in is rather minimal, so it's very, very thick. Right? So that's dense connective tissue. Right? Now the fibers found there can either be considered regular or irregular. So regular means that, let's say, the spaghetti and the collagen fibers and things that we're looking in there are very parallel with one another. They're densely packed in there and very parallel, kind of like a box of uncooked spaghetti. If you have a little plastic window where you can look into, the interior of a box of spaghetti when you buy it, and you'll see all the spaghetti in there. They're parallel, they're vertical, they're visible, and they're packed in there without a lot of space in between them. That would be considered regular. If I took that same box of uncooked spaghetti and I poured it out onto the countertop, they kind of have a tendency to fall in different directions. Now, instead of being all parallel with one another, they're kind of coursing in different directions. That would be irregular, an irregular orientation of the fibers in the cells. So when we look at, when we start looking at things like even skeletal muscle compared to smooth muscle, one of the features of it is that it has a very regular and parallel appearance to the fibers. That's what we're talking about. Other connective tissues include our cartilages, which we'll look at when we cover the skeletal system. We have 3 types of cartilage: hyaline, elastic, and fibrocartilage. And the cells that form and maintain cartilage are called chondrocytes. So chondro meaning cartilage and cyte meaning cell. Another category of connective tissue is bone that I just described. Before we were talking about, I think it was in the last lecture, perhaps, we were talking about bone just briefly. Compact bone versus spongy bone, because the spongy bone is where we have red bone marrow and all blood cells come from there. All right. So we have bone, which imparts certain function. Most of what we think of is nice compact bone that forms the long and short bones of our body, but within there is those layers of compact bone, we have spongy bone, and that's where we have red bone marrow, and we have the hard substance of spongy bone called trabeculae. Right, so bone has its own kind of category within the connective tissues itself. And then we have blood. Blood is the only liquid connective tissue. We have formed cells within blood and plasma is about 55% of your blood, and out of that 55%, 90% is generally water, 90 to 92%. So it's a very liquid form of connective tissue. Right, so here we have some different pictures of connective tissues. Up to the upper left-hand side, you see how these fibers are coursing in all these different directions, and then you can see some nuclei of some kind of cells in there. Those are generally fibroblasts. But you see how you have these fibers coursing randomly in all different directions? I call that the Jackson Pollock painting appearance. You have some very thin, dark staining pink to purple fibers. And then you have some larger diameter ones. When they're coursing by themselves, you can they have this like translucent appearance. So these that are not dark purple, pink staining are the collagen fibers, and they're rather large diameter. You can see a few here and there. The little thin, very skinny little fibers that you're looking at that do take up a darker, fuchsia, pink, purple, whatever this is, those would be elastic or elastin fibers. That is a lovely picture of areolar connective tissue. It's a dense connective tissue, but the fibers are irregular and it has an irregular orientation. Over to the upper right-hand side, here we have what's called dense regular connective tissue. Dense regular connective tissue is composed mostly of collagen. And you see how it kind of looks like these wavy lines, but they're all stacked on themselves, and they're all rather tightly layered, fibers of collagen to collagen to collagen. So we don't see a lot of fluid accumulating between these layers. It just looks like a rather solid sheet, of these fibers that are very parallel. That would be dense regular connective tissue. And then down at 3 o'clock, that is blood, where we have all these red blood cells or erythrocytes in the background, and some of those larger cells with the big purple nuclei in them. Those are 2 of our white blood cells, 2 of 5 white blood cells. A lymphocyte and an eosinophil. Very good. So that's our only liquid connective tissue. Then down on the lower right-hand side, that is compact bone. We're looking at one particular osteon. We will look at these again when we get to the skeletal system, but it kind of looks like a tree you cut down and rings of a tree with a little core in the center. That is one repeating unit of compact bone called an osteon. Right. So that would be compact bone. And then over towards 6 to 7 o'clock, here we have an example of elastic cartilage. you can see these blue kind of purple fibers that are rather thin. Thus, it's elastic cartilage because that's elastic or elastin fibers. And you see those little, I don't know what they look like, eyeballs almost floating around. All right, that in the interior of one of those kind of white little sockets or rooms you're looking are the chondrocytes, the cells that form all this cartilage. So that would be an example of elastic cartilage. Most of the examples that you're going to look at on slides, either in the lab here or in the textbook, will generally have a lot more elastic fibers there, so that in the region of fibers in between any of those chondrocytes sitting in there, what's called the lacunae, or the little room that they're sitting in, will be very dark purple and contrasted to those cells sitting in their little room there. And so you have one identify is that sharp contrast between dark purple fibers, stringy fibers that you see, very abundant, and then you can see a chondrocyte sitting in its lacunae. And when it's in that little room or lacunae, there's often a lot of fluid in there, so that's very pale staining compared to the dark staining fibers adjacent to it or outside of it. And then lastly, about 9 o'clock, that is fat tissue. It's called adipose connective tissue. Each of those kind of pink, circles there is one fat cell called an adipocyte. And then you just have a little bit of connective tissue in between. So fat tissue or adipose connective tissue, have what's called, we consider either like a chicken wire, if you know what chicken wire looks like, a chicken wire appearance, or maybe a honeycomb appearance, where the contents of the interior of this adipocyte is mostly fat, because that's what it's going to store. And its nucleus is often pushed way over to one side or the other. So sometimes you can see it squashed there. Sometimes it's actually on the back side of the cell. But the majority of what you're looking at, the interior of that cell, is fat being stored in a great big vesicle. And fat doesn't take up stain well. Usually the stains that you're going to use to stain structures will either have a positive charge or a negative charge to it. And since fat is non-polar covalently bonded, it just doesn't take up stain well. So these cells that have this, cavity kind of region in the center of it, and that they repeat all over and over, you generally don't see one, you see many of them. That is adipose connective tissue. Okay, then we move on to muscle tissue. Muscle tissue contains elongated cells called fibers, most of them. Smooth muscle, they're not very elongated. They kind of have what's called a fusiform shape, which is like a cigar shape. But skeletal and cardiac muscle have rather elongated cells. They use ATP to generate the force to produce movement. So muscle tissue is designed to shorten and lengthen, shorten and lengthen, shorten and lengthen, many, multiple times within the muscle tissue to get any real movement occurring. So they're going to use a lot of ATP, which means they're going to have a lot of mitochondria, right? They are highly vascularized because to produce ATP, we're going to need oxygen. So you're going to have a very good blood supply to deliver oxygen and a substrate to burn in the mitochondria, optimally or preferably glucose. So we have 3 types of muscle tissue: cardiac muscle, skeletal muscle, and smooth muscle. And these are pictures of them. So, the cardiac muscle, we're going to come back to, in the cardiac system or the heart chapter in AMP2. And smooth muscle, you see this kind of tissue, found in multiple different organs and things. And generally by location of where you are, if it's muscle tissue that forms this muscular layer, thus it's going to be some kind of muscle tissue, then it's usually going to be smooth muscle, except if we're talking about the specific muscle in the heart, which is cardiac muscle tissue, or maybe the muscle that's under volitional control, like the muscle that attaches from bone to bone that you can contract so you can lift a book or stand up or walk or sit down. That's going to be skeletal muscle. So smooth muscle is found every place else that skeletal muscle and cardiac muscle is not. And cardiac muscle is only found in one place, the heart. All right, so in the upper left-hand corner, B here, that's pointing to the nucleus of a smooth muscle cell, but that's an example of smooth muscle. They have what's called more of, like I said, a fusiform shape that kind of looks like a cigar. And then to the right, at 2, 3 o'clock is skeletal muscle. And each one of these parallel, it looks kind of like a straw, but that is one skeletal muscle cell, right? And they're stacked on each other. They have many nuclei, and that's another feature of skeletal muscle, is that it not only has a lot of mitochondria, it's got to be multinucleated, and they're stacked on itself. So they're very parallel. They may be at an angle, they're kind of coursing in an oblique angle right now, but those are very parallel muscle cells. And that is so when this muscle shortens and contracts, it approximates the two ends of the muscle so that we approximate the origin and insertion, and we get two bones to move. And then at the bottom, we have cardiac muscle tissue, very, rather similar to skeletal muscle, but it has more of a branching effect. The cells are very parallel. They're rather parallel to one another. They're very linear, but they have a tendency to cross on themselves. So it's kind of like what I refer to as a bamboo appearance. If you have bamboo in your yard, or if your neighbor does, or somebody does, that each shoot of bamboo is very linear, but they, grow upward toward the sky, of course, but they kind of cross each other, and that to me is what cardiac muscle tissue resembles. Okay, nervous tissue, contains many cells and is also avascular. They function to monitor and react and form a electrical kind of communication network within your body. We have two types of cells that are found in the nervous tissue. And we're going to come back to this again when we get to the nervous system, which follows the muscular system. We have neurons and neuroglia. So they all have kind of nervous something in its title. Neurons, of which we have three basic types, are able to generate and conduct some kind of an electrical impulse, maybe a short distance, maybe a long distance, maybe from your neck area, from your cervical spine to the muscles in your forearm or in your hand. They can travel long distances, or they may travel short distances, but they conduct these impulses. And so they have certain characteristics associated with their functions. Neuroglia are supporting cells, or cells that support and insulate and protect and help those neurons do their job. We have 4 different classes of neurons found in the central nervous system, and then we have 2 classes of neuroglia that are found in the peripheral nervous system. But they don't conduct impulses, but they have other functions, and they help these neurons do their job and help the nervous system do their job. This is a picture of a multipolar neuron, right? And unipolar neurons and bipolar neurons, have somewhat similar, but kind of different shapes to them, but have this very unusual shape. So when we're talking about cells, in the previous chapter, I was saying that the cell model, that composite cell model, has that planetary model, depiction all the time. They're rather round with a nucleus in the center and the little organelles, around it. And I kept saying, there aren't that many cells that are like really round like that. They're really unusually shaped. And this is one of them. This is a multipolar neuron. This is one cell with, you can see like a darker staining, pinkish purpley nucleus with a nucleolus in the center, actually, and then we have some other what's called Nissl bodies kind of thrown around in there. But we have the cell body of this neuron. You have these little extensions, kind of look like braids that extend from it, called dendrites. And then one of those, which I believe is probably to the bottom, like around 7 o'clock, is going to be its axon. And this cell is capable of responding to different events around its environment, generally different neurotransmitters. And then it generates a signal based on the stimulation it receives from other neurons, and then sends a signal down this axon to whatever organ, or cell or other neuron it needs to communicate with, in a chain of, one cell to another, trying to communicate and send a signal of some kind. And that is clearly not round. So I think those are really interesting to see, because then you start to realize, wow, the cells of my body really have all these vastly unique, and wild shapes, associated with them. Okay, so that takes us to the integumentary system. All right, so we just looked at like tissues, CMEN, over and over and over again, semen, connective tissue, muscle tissue, epithelial tissue, and nervous tissue. Going forward for the rest of your life, any tissue you look at will fall into one of those four categories. And as I said before, the muscle tissue only has, three types of muscle cells. So that's not too bad to start to memorize now. The nervous system has just the neurons and neuroclia. That's not bad. The epithelial tissue, okay, it's found in certain regions and that has that naming system. Simple, stratified, pseudostratified, maybe the first term, the number of layers, and then the shape of the cell, squamous, cuboidal, or columnar. And then its last name, which you theoretically should be naming, in lab and any place else, is epithelium or epithelial tissue. It's a very proper thing, how you name tissues. And it's the connective tissues that seem to have the most diversity. Fat tissue, blood, dense regular connective tissue, dense irregular, dense irregular connective tissue, areolar connective tissue, cartilages, high and elastic fibro, bone, compact bone, spongy bone. There's a lot more variety. So I guess when in doubt, if you don't know what it is, throw it into the connective tissue category. But after a while, you get this, you're going to get the muscle tissue over and over again, epithelial tissue all over, especially the AMP2. Okay, and in the integumentary system. All right, so the integumentary system, the organ, is skin. We have different tissue layers. Technically, the skin is only composed of an epidermis and dermis, but we put it into layers, and the three layers that we find in the area of the skin, I guess you could say, are the epidermis, dermis, and hypodermis. Right. The most superficial layer is the epidermis on the top. The dermis is in between, and hypo means below. So that's the deepest layer, otherwise referred to as the subcutaneous layer. Right. It also includes appendages like hair follicles and hair that grows off the surface of your body, nails, exocrine glands called, the sweat glands and the oil glands, or the sebaceous and sudoriferous glands. They all develop from the epidermis and maybe get pulled down a little bit, and down into the dermis. But I'll show you how you can tell that they're still part of the epidermis, but they kind of get pulled down like roots growing from a plant, down into the soil. It's highly innervated, so you are able to monitor the external environment. So here we have a picture of the skin. This is from your textbook. And, don't quite look at all the words on the side, because then you get kind of confused. You go, oh my God, this is like the busiest diagram I've ever seen. But you take it in pieces. So the way I approach this is, without looking at all the terms to the left and right and to the bottom of this image, is that we just look at the layers by color. You have kind of a brown layer, you have a pink layer in the middle, and then you have yellow on the bottom, right? All right, so that's it. From the beginning of the brown, or everything in brown is part of the epidermis. And everything in the pink layer is the dermis. And then once you hit the yellow layer, which is fat, then you're in the hypodermis or subcutaneous. All right, so we have layer, layer, layer. And these layers, they're given different models, maybe, use different colors, but it's all the same. So if we look at the epidermis, we can see the surface where we have hair growing out of the surface. And then over to the left-hand side, we have epidermis with a little bracket there. So it's including that brown tissue. The inferior border of that brown tissue, look and see how it undulates like waves on the ocean, and then look how it envelops and wraps around this hair follicle, because that's a hair follicle. All right, the hair grows down inward. It pushes down like roots, into the dermis layer in pink, but it is part of the epidermis. Attaching to it on the right and left will be oil glands. Think oily hair, because that's how oil will be liberated. It'll travel along the hair shaft and to the surface of the skin, so it follows that tube system. And and so forth. So you can see how each hair kind of digs down, hair follicle, and the epithelium that surrounds it digs down, kind of, it extends down into the dermis, but is brown. So that trying to tell you color coding, that's part of the epidermis. Right, then we have the dermis, which is the pink layer. Now that has a lot of stuff in it. So, starting over on the left-hand side and working our way across, they identified the most superior layer, which is about the upper 20% or so of the dermis, is called the papillary layer. And then everything else below it is considered the reticular layer. Next to it, we can see some blue vessels and red vessels. So here we have a blood supply. And note that the blood supply terminates in the papillary layer there, the upper layer close to the epidermis. It does not extend into the epidermis. That's why we said that epidermal layer is avascular. It lacks a blood supply, and the epithelial tissues lack a blood supply. And that's kind of a protective mechanism, because the blood vessels, if they extended farther toward this, any farther really to the surface of your skin, it would be very easily compromised by bacteria and viruses kind of trying to squeeze between, those squamous cells. Or when you had the very smallest little cuts on the surface of your body, those bacteria would open up a route of infection, and they'd be much closer to your blood supply. So you really want to keep a nice physical wall like that between the external environment and the internal environment. But those squamous cells will receive via diffusion a lot of oxygen and nutrients and things, because these are capillaries at the superior area of the dermis. And now we have the ability of different gases and nutrients to leave the blood supply and diffuse to those squamous cells, that are growing above them. Right, so we have a blood supply. We have different nerve structures down there. We have about midway across between that one hair shaft and the other. You see that funny look, it's called a lamellated receptor here. That would be a Pacinian corpuscle. Those are generally Pacinians that have those, kind of looks like you cut into an onion and you can see layers of an onion. That is a type of a pressure receptor. We have other nerves, that and receptors that are found here that extend way up to the top. These looks like little yellow, a yellow tree growing with little finger-like extensions that appear to be stopping at the epidermis, but they actually extend a little bit up into the epidermis here. Those are called free nerve endings. They act as pain and temperature receptors. We have some, I'm not sure why they don't identify them here, because I think it's easy enough to do, but you see the little muscles that are attaching to those hair follicles? Those are called the erector pili. Those are the muscles that contract and cause your hair to stand up on end. I kind of like those little ones, erector pili. It's kind of fun term to pronounce. And then a little farther over, you see this, it looks like a coiled hose that extends in brown up to the surface of the epidermis. Right, that's a sweat gland. Oil glands will use the hair shaft, and follicle to deliver oil to the surface of the skin. But the sweat glands, right, like looks like a coiled hose, that is a gland. The duct is the single hose that travels up to the surface and then liberates sweat on the surface of your body. Right, so that's the majority of structures that we're finding kind of uniquely in the dermis. There are different types of receptors, or pressure receptors. We have Pacinian corpuscles and Meissner's corpuscles, but we'll look at those, when we look at some of the things, that are the particular nerve structures found in the dermis and hypodermis. And in lab, we have multiple models. Some of those big cookie sheet models, I call them big plastic models, that look like a great big cookie sheet, that show these same layers, but in different colors, and the same structures, just in different colors, in layer to layer to layer. And then we have some that are, they're not a great big model, but they're up at about 12 inches high by about 18 inches wide, and they're like on wood, essentially, and there's smaller models that represent similar structures. Okay, so tissue layers. We have the epidermis, dermis, and hypodermis, or subcutaneous. The epidermis, we find stratified squamous epithelial tissue entirely. Now, there are some, we're going to talk about some of the other things that are found in the epidermis, but we have some other cells, thrown in there, but the primary tissue is stratified squamous epithelial tissue. The dermis, we find primarily dense irregular connective tissue, which is the majority of the background there. Let me just go back a picture. You see all the pink in the background? Right, that represents the dense irregular connective tissue. But we have, some other tissues found. They're like simple cuboidal epithelial tissue, because that's what forms the glands, like that sweat gland that's by itself that looks like a coiled hose. And then we have in the hypodermis, we have primarily adipose and areolar connective tissues. The fat tissue, and then the areolar connective tissue is that Jackson Pollock painting type of a tissue we looked at before that has collagen and elastic fibers, that help connect everything above it, to the muscle layer that will be, now inferior to it or deep to it. And it's capable of being stretched in multiple directions and bouncing back again. Okay, so the epidermis, its general function is to provide a physical barrier between everything outside versus everything inside. It helps protect from dehydration, injury, and infection, and it's avascular. And so remember, the blood supply, those capillaries, those red and blue vessels stopped in the dermis. The most inferior point of it, where the epidermis meets the dermis, where you have that first region of brown in the last picture, adjacent to that pink, that is called the stratum basale. You have multiple stratum, of the skin, and essentially you're taking the epidermis and creating and identifying more layers within it. And so, the only one I really want you to know is the stratum basale, which is at the base of that layer. Right? And that is where we have high mitotic division of squamous cells, because it's from there. And let me just go back again so I can show you that picture. All right, so let's look at the bottom where the brown meets the pink. You see where it becomes slightly darker or tan or brown at that point. That is your stratum basale, where the brown meets the pink. And that brown stratum basale is where these squamous cells will go through that interphase, prophase, metaphase, anaphase, telophase rapidly. Why? Because from that basement point, it pushes all those cells up toward the surface where the hair is exiting the skin. And that's where the cells will eventually become dead, and will just exfoliate off. Well, you have to keep replacing them from the bottom and just keep pushing them up, like pushing your hands up toward the ceiling. And so that area, the stratum basale, is very important, because that's where we have the high mitotic division area. Okay. Other cells found in the epidermis are keratinocytes, especially found on the palms of the hand and on the soles of the feet. We have keratinocytes, and they produce keratin, which is essentially a waxy substance. It's kind of a waterproofing layer on the palms of your hands and the soles of your feet. Because millions of years ago, we didn't have a lot of shoes and things, and you used a lot of tools on the palm side of your hand. And so it helps protect it. It helps keep fluid, because keratin or wax, all right, wax. So think about wax. How soluble is that in water? Not. All right. So it helps seal, like moisturizer that you put on your skin, the fluid inside your skin from evaporating away. And so it helps protect it then. Melanocytes are dispersed in the epidermis as well. They produce melanin for UV protection. We have dendritic cells, which are a type of white blood cell called a macrophage, and they help to, phagocytize or ingest different microbes that might be passing through the skin, which is handy, because even though that's, stratified in its multiple layers, it's not like some of these organisms can't, for one reason or another, squeak through there. And usually it's because you have like this minor little cut, or even a major cut. But I mean, you have to have a cut in that layer, and that becomes a route of infection, like a crevice in your skin. And so to keep those bacteria or viruses or parasites from getting any further, toward your blood supply that's in the dermis, you have these cells that can phagocytize them, break them down, initiate your immune response. maybe inflammation there to help, inhibit any microbial invasion. And then we have cells called like tactile cells that provide some sensory reception for touch. Those we don't really look at, with the exception of part of that free nerve ending that extends into the epidermis. There isn't one a clear example of that on the model. So we got some new model that I'm not aware of. Okay, so let's go to the next layer, the dermis. It contains a lot of receptors, such as the free nerve endings. They look like a tree that's growing into the epidermis. Meissner's and Pacinian corpuscles, we're going to look at specifically what they do, but they have particular shapes. One is found closer to the epidermis, one is farther away or deeper, and embedded in the hypodermis. We have oil glands and hair follicles that grow downward. So they're within visually in the region of the dermis, but they're not technically part of the dermis. They're part of the epidermis. All right, so we have free nerve endings. Free nerve endings detect pain and temperature, or what's called pain and temperature receptors. Receptors are nerve structures, not cells, but they're a structure, like the thermostat on the wall or in your office or in your home that detects the change in temperature. All they do is detect a change. And in doing so, they can help send a signal on what's called a sensory neuron to your central nervous system to let you know that maybe you put your hand down on something sharp and now is creating pain because you damage cells, and that's what stimulates pain receptors, or you put your hand on something very cold or very hot. Meissner's corpuscles or tactile corpuscles, detect light, pressure, or touch. So light, pressure, and touch, because these Meissner's corpuscles are closer to the epidermis and toward the surface of your body, it doesn't take much pressure to reach them and then stimulate them. So if you have light pressure that is just reaching this one receptor and not receptors deeper to it, then your brain interprets the signals from there going, okay, this is just something that's very light on my skin. Maybe it's just, a gnat or a mosquito landing on the surface of my body, maybe on a hair, and you're bending the hair and that lets you know something's on the surface of the body. Maybe it's just some light clothing, on the surface of your body. Pacinian corpuscles are lamellated. They're the ones with those rings, when you kind of cut it open, you look into the interior, it kind of looks like layers of an onion, right? They're found deeper in the tissue. Therefore, they respond to deeper, more profound touch or pressure or maybe vibrations, because you have to displace everything in the epidermis and part of the dermis before you even reach these that are deeper. And then now that you're stimulating both the Meissners and the Pacinians, because generally you hit the Meissner on the way to the Pacinian, is that now all those signals are traveling to your brain, and that's saying, hey, something much heavier is on the surface of your body now. The dermis is composed of, as I described before, the upper 20%, which is called the papillary layer, is a thin superficial layer made of highly vascularized connective tissue. And then we have the remainder of it, 80%, predominantly dense, irregular connective tissue, that gives strength and elasticity. It allows the skin to stretch without tearing and kind of bounces back again. So this is a pretty good example. Our slides don't really quite look like that, but the epidermis looks more of that. I'd say the dermis doesn't generally take up that kind of dark purple, pinkish kind of a stain, but the epidermis can kind of look like that. The dark purple undulating point there, that is your stratified squamous epithelium. And you see that darker pinkish purpley layer on it? That's the keratinized layer. We find that very thick on our palms of our hands and on the soles of our feet. But if we looked at the skin that's on like the inner surface of your arm, let's say, like on your forearm, or an inner part of your biceps, you wouldn't have that thick keratin layer, but you do have that in certain areas of the body. But anyways, that would still be considered part of the most superficial layer, the epidermis, and the dermis below. They're showing you the papillary layer, and then everything else would be the reticular layer. We can see some other connective tissues in there. We'll get to another slide, I think, that lets us blow that up a little bit more. Okay, and that takes us to the hypodermis or subcutaneous. Technically not part of the skin, and deep to the dermis, but we include it in these layers anyway. Contains many larger blood vessels because the blood vessels enter larger, in the hypodermis, and then they'll narrow down, to be smaller and smaller ones, as they approach that papillary layer of the dermis, and where they're going to discontinue, they're going to go up, and then they're going to come back down. So it contains many blood vessels, nerve endings, adipose and areolar connective tissue. Adipose connective tissue stores fat. So think, when we talked about lipids in the chemistry chapter, same functions: long-term energy storage, cushion and insulate organs, shock absorber, all along the same lines. And areolar connective tissue, because it has so many collagen and elastic fibers, is it helps anchor the skin to the muscles, and it can, again, be kind of stretched and bounce back again. Skin appendages, we have hair follicles. They are keratin strands that function as touch receptors. If you just have even the smallest little gnat, land on one of those little fine hairs, maybe on your arm or on your leg, you are aware, generally, unless you're very, very busy and distracted with something else, you're generally aware of it. So they act as a touch receptor. The skin, hair, depending on how thick and where it is in the body, can help retain heat and act in UV protection. Nails are scale-like modifications of the epidermis that function for grasping small objects, protection for the ends of your digits, and for scratching. And then we have exocrine glands. We have our sweat glands and oil glands. Sudoriferous glands are our sweat glands. And that sweat helps regulate body temperature and protects against infection. You not only use the watery component of sweat to evaporate off the surface of your body, take heat with it, and thus cool the tissues of your body, but also within sweat, we have salts in there, and it's rather acidic. So that in and of itself sort of creates an inhospitable environment for some bacteria. You want to create environments to kill bacteria, that they don't withstand very well. They can't really survive, or they cannot proliferate at least. And so that's one of the mechanisms. Sebaceous glands do the same thing. They help protect from infection as well, but they also help because oil doesn't evaporate, is that it puts kind of a coating on the surface of the skin, and so that you slow down the evaporation rate. All right, so that takes us to Kahoot. So for those of you who would like to play, I'm going to change my share, so bear with me. Let that boot up. And let me get to the point right there. It is. All right. I'll reshare now. Move this over here. There we go. All right, is everybody in that wants to get in? Even if the quiz gets started, the pin is always down to the lower right-hand side. So I'll give you another few seconds and then we'll get crack-a-lacking. Okay. Very good. So the Meissners are closer to the surface. The Pacinians are a little bit deeper. The Meissners, when you're looking at, I don't know how many people are going to be on campus and kind of doing this little face-to-face, or I'm pretty certain that a lot of the labs that are even online from this campus are using some of the pictures of the models we would have used, or that we do use in the lab. And those little Meissners, kind of look like a little sperm. It looks like it has this body with a little kind of a nerve tail to it. So I say, don't touch that sperm. So you know that it's a light touch receptor. The Pacinians are found deeper. They're the ones with the different layers to it, like an onion. So the closer you are to the surface, the less pressure it requires to reach you and squish you. I mean, because these are types of receptors, mechanoreceptors, that something physically is happening to stimulate it. So you are actually performing some kind of pressure and moving it, making it bend, or compressing it to stimulate it. Unlike a photoreceptor of the eye is just responding to these light photons. I mean, it's like they're energy waves more, or chemoreceptors respond to the pressure, the presence of maybe hydrogen ions or oxygen or carbon dioxide or, different chemicals in the environment. But mechanoreceptors have something physically like happening to them, and they're in that class. Very good. Epidermis, dermis to hypodermis, or subcutaneous. All right, pretty good. The epidermis, that's where we find simple squamous epithelium. Then in the dermis, we have mostly dense irregular connective tissue, and we also find some simple cuboidal epithelial tissue. Actually, a good amount of it because you have a good amount of sweat glands. But that wasn't an option. Maybe if I wouldn't give you like two correct answers, or if I did, then I'd say A and like option D might be A and B, something like that. But areolar and adipose, A and A's, are found in the hypodermis subcutaneous primarily. Or maybe regulating body temperature. I don't know, maintaining body temperature. Yeah. Okay, good. Good. Very good. The blood supply, and that's, again, like when we get into AMP2 and we're looking at the immune system and the ways, what we call first line of defense, what are the first things that is our physical barriers of different types, like mucus and your skin, that act kind of like a plasma membrane, to keep everything outside that needs to be out, and, and protects everything inside or on the other side. And so that's your epidermis, and you don't want that to be highly vascularized. Good, both ANAs. They're in the adipose and areolar connective tissues. Nice, very good. Your hair doesn't sweat. You might have sweat on your hair, but it's not from your hair. Right. It serves a lot of different functions. Acts as like a touch receptor, kind of a pressure receptor. It helps protect from UV. It helps insulate the body. So yeah, good. Very good. All right, very nice. Let me break that share and close that out. Okay. Oops. I've got to find you guys back again. Hold on. I've got to change my share real quick. I have to go back to my notes. Not too much more material. Let me just quick go. Okay. Sorry about that. Move that in this over a little bit. Oops. Okay, can everybody see the notes?
Yeah.
Okay, just making sure because I have a couple other pages open. So, all right. Okay, so here are some functions, general functions of the integumentary system. Well, it acts in protection, and actually it's a really good protector on the most part. It surrounds your whole body. It's kind of like a plasma membrane. It regulates the passage of material across it, from what's outside the body to inside the body. It functions in thermal regulation. So, with sweat glands and things, it helps cool the body when necessary. It provides cutaneous sensation, which means we have different receptors close to the surface of the body that detect changes that will help keep you safe. I mean, you need to have these pressure receptors and pain and temperature receptors so you don't like burn the skin off your hand, because that creates an open route of infection, and not only is it a route of infection, but extremely damaged tissue would not allow you to use maybe your hands or feet or something for a period of time, and therefore maybe now it just means a vacation, either you stay home, you take a week off from school or work or something, or 2 weeks off, but millions of years ago it means you'd probably die. You wouldn't be able to survive. All right, metabolism. This is where we produce vitamin D. What is it's the inactive form of a hormone that we use to help increase blood calcium levels. It acts as a blood reservoir, which means we can store some blood there. And it functions in excretion and absorption because, we can excrete certain products, and things through sweat. All right, so as a protective barrier, it functions as a chemical barrier. The pH, and I was mentioning that before, the pH of sweat is rather acidic and acts as a bactericide, or just antimicrobials, probably easier to say or think. Oil contains an antimicrobial as well to inhibit bacteria. Melanin, helps prevent UV damage. So physical barrier, the stratified cell layer of the skin, the nature of it, and then especially to the palms of the hands and soles of the feet, that keratin, the thicker keratin layer, make it more difficult to penetrate. And then we have a biological layer, some of the other cells that we find there, like the dendritic cells, that are white blood cells that can engulf and phagocytize invading microbes and stimulate the immune system to let that you have an invading organism and you can mount what's called an immune response. Thermoregulation, regulated by a negative feedback mechanism. What happens when you have a higher than normal body temperature? What happens when you have a lower than normal body temperature? Well, when you have high body temperature, living in Florida, virtually everybody knows this, is that the vessels will dilate that carry blood to the surface of your body, in your dermis. All right. So we're going to bring more blood to the surface, which helps to release heat. And to accentuate that, we secrete sweat. And when water evaporates and it goes from a liquid form to a gas form, it takes heat with it. And it takes a good amount of heat for water to go from a liquid to a gas. Right. So, that's why you feel so much cooler, so much quicker if you're sweating and you stand in front of or below a fan, because that moves the air across your skin very rapidly. It causes the water to evaporate more quickly, and thus you feel a very rapid cooling sensation on the surface of your body. Thank goodness, right? Because it's pretty darn hot out right now. Low body temperature, the opposite. We have the opposite reactions. We have blood vessels that constrict, and thus we kind of route blood back to the core of the body, so bypass the skin, retaining heat for internal systems. The adipose tissue, the fat in the hypodermis subcutaneous, helps to retain heat. Hair follicles also act as an insulator. And then one of the other responses is actually if you keep getting really cold, you'll get to the point where you're shivering. And that skeletal muscle contraction and muscular contraction, we said one of the functions of the muscular tissue was to generate heat. That's an example of generating heat. We don't often shiver too much here in Florida because, well, I'm from New York, so it gets cold sometimes, but it doesn't get like the kind of bitter cold I've experienced before. But it can get pretty cold. But I don't think I've ever really shivered outside here, ever. But for you guys that are born and raised in the South there, you a little more sensitive. All right, cutaneous sensation contains receptors that respond to stimuli outside the body. Free nerve endings detect pain and temperature changes. Meissner's corpuscles are the little ones that look like sperm, so don't touch that sperm. They're the ones that are found, toward the surface of the body or more closer to the epidermis than the Pacinians are. They detect light touch or light pressure. Pacinian corpuscles are found deeper than the Meissner's, so they respond to or detect deeper, degree of touch or pressure and vibrations. And hair follicles help with cutaneous sensation because they detect, touch or light touch on the surface, movement such as insects. So that's how we're kind of regrouping some of these things together as to whether they function, the things that we talked about, are they in protection, they're part of chemical, physical, or biological protection. When it comes to thermoregulation, it's really just sweat, but what might the body's response to be when you're, higher than normal body temperature or lower than normal body temperature? And then what are these things, we group them together, hair along with those different receptors as to, what are the different receptors that respond to some kind of changes in the external environment, some kind of skin sensation. Vitamin D synthesis. So UV stimulates skin cells to produce vitamin D, which is the inactive form of what's called calcitriol. When blood calcium levels fall below normal levels, the vitamin D will be converted to calcitriol in your kidneys. And that, is stimulated by a hormone called parathyroid hormone, or PTH. And we'll look at that again, when we look at the endocrine system, which is part of a really exam 2 material in EP2. But calcitriol, once we convert vitamin D to calcitriol, then calcitriol travels to the intestines and stimulates absorption of calcium from there. And thereby now, the more calcium we absorb, we increase the calcium in our blood. We increase blood calcium levels back to normal. Boom, now we have another example of a negative feedback loop. So when blood calcium levels fall, right, we will then convert vitamin D to calcitriol. Calcitriol targets the small intestine, increases calcium absorption. Calcium comes back to a normal level, and now you stop secreting, the parathyroid hormone. The reason, we keep all this vitamin D around so that when we need it, and when it comes to at least calcium absorption, not that vitamin D doesn't have other functions, because it does have some, and they're finding probably more vital functions than we ever thought before. But as far as affecting blood calcium levels, we want to have the vitamin D there, but we don't always want it in an active form, because we don't want to keep absorbing calcium from the small intestine if it's in a normal range, or if it's even higher than normal, because now you have too much calcium. Now you have to find a way to get rid of it. Right. So the vitamin D just kind of hangs around, circulates to the body. We keep creating more, in the event that, as I said, it performs some other functions, but when needed, when the calcium drops too low, now we're going to turn it into a molecule, that is an active form, a hormone that is an active form. And now we increase the calcium absorption. And then when that reaches certain normal levels, or the calcium reaches normal blood calcium levels, then we stop secreting the hormone that converts it. And now we just leave the vitamin D, whatever is there left is just in the form of vitamin D until we need it again. And it acts as a blood reservoir. So we probably, plus or minus around 5% of the body's blood circulates through the skin like that. If you're cold, you're going to vasoconstrict. Less blood is going to be there. If you're hot, you're going to vasodilate, and a little more is going to pass through there. But it's about 5%. Excretion, we can lose a lot of salts and water through sweat. Sweat, there are normal ways in which, or methods in which, we lose fluid from the body. One of them is through creating urine and excreting it from the body. But we lose fluid also from just the action of inhaling and exhaling, and taking dry dry air from the external environment and moisturizing it. So when when you breathe in, it becomes moisturized as it passes through your nasal passages and your sinus cavities and along your trachea and so forth. There's mucus there, and that helps moisturize and cleanse that air before it reaches the smaller regions, the microscopic regions of gas exchange in your lungs. That's the way we lose water. We lose water through sweat. We lose water through tears. We lose fluid, and replenish fluid in serous the serous fluid between serous membranes that lines the digestive system, that needs to replenish and circulate through your connective tissues, that provide the fluid, for synovial joints. Those don't turn over quite so much, but anyways, cerebral spinal fluid, maintaining appropriate amounts of cerebral spinal fluid. So we use sweat and water for multiple things. Generally sweat, if we have a more than normal hydration level, or being kind of hyperhydrated, so to speak, it's not like all of a sudden you're going to profusely sweat to decrease the amount of water circulating in blood, but we do lose water that way. We don't do it often intentionally. It's kind of by the cooling effect, really. We also can excrete small amounts of gases like carbon dioxide. We can get rid of, ammonia and urea, that way as well. So there's a certain amount of excretion, occurring there. Absorption, not that much. We absorb light photons there, and thus melanin works really well. Substances can be absorbed. There's a lot of research that will contradict, the efficacy of a lot of these really expensive moisturizers that you can put on the body, that it gets absorbed and it changes your skin dramatically. It only, coats to and is absorbed to a certain point. Although some toxins, some different gases, can definitely be, absorbed a little bit more, and deeper, especially if it's in large amounts or certain chemicals on the surface of your body, but not that much absorption, mostly excretion. Okay, so that takes us to, I believe, what is going to be our last Kahoot. Let me get logged in. There we go. All right. Very good. Nice. Good, good. Yeah, that's an easy one to remember because they're all kind of like neuron, nervous, neuroglyph. They're in the same category. Good. Yeah, that's the Jackson Pollock painting, I call it. That's the areolar connective tissue. All right, well done. Hey, Levi, you're able to hold on. Last time you got kicked off the podium right at the end, or just about at the end. Good job. Okay, so let me just check this. Yep, that's the end of the presentation. Let me just make sure. Yep. Okay. Ah, did that just close me out? Okay, so this sometimes I like these Zoom things, but the funny things happen sometimes here. All right, so does anybody have any questions? That's the end of the material. Your exam is next week. We generally Zoom, live Zoom on Monday, Wednesday, and we don't have any class on Monday. So instead of just having the exam open just on Wednesday, I'm kind of giving you the whole week. Let me just check to see if I scheduled anything else for you guys. Hold on. Not really. Right, so, I had designated next Wednesday to be, just for you to review and study or whatever. What we can do is if anybody's interested and you can let me know, is that I can live Zoom for, at 5:30. I mean, doesn't really matter. I can live Zoom with you if you want to do, go through the other Kahoots. I guess, if I hear from someone that wants to do that, that's fine. Otherwise, I'm just going to leave you be and let you just get the exam completed. But if someone's interested in doing that, honestly, if you go back through the Zooms that are uploaded there, you see the same ones anyway, so you don't have to wait for me. And yeah, actually, I think I'm going to defer to that direction because that way I can spend that night putting out fires. The Kahoot quizzes, if you download it and you look at the Zooms that I uploaded, if you just advance quickly through them till we get to where a Kahoot quiz is being taken, you can just look at it that way, and that you don't have to wait till next Wednesday. So maybe that's just a better plan. Just look at the Zooms and advance it, rapid advance to where one of the Kahoot quizzes have been taken, and then you can review it on your own. And that's probably like way faster than, waiting for a week from tonight. And if you do that, then it's like I would try to cut yourself some slack and get that exam done earlier than later and just check it off your list because it's, when you don't do things quickly, often all of a sudden you get to Saturday, Sunday, and you go, oh my God, I didn't, I forgot about, and then there's a storm or something, or something happens to your computer or whatever. So you really need to plan ahead, and, make sure that you, are prepared. But then you get a good, 5-6 days off a week before the next, possibly, until the following Monday where we have another lecture or Zoom. And then we'll start the skeletal system, at that point. So I know no one wants to spend a lot of time over the holiday weekend, but I don't think it's really terrible quantity. I know the chemistry seems to be a little overwhelming, but now that you're getting your labs done and the labs are kind of catching up as well, is that you should be able to go back and really restudy the introductory lab and the chemistry lab, and it should really now be coming together. And that would be the first thing I do, is then and then jot down any questions you might have at that point, and either ask your lab instructor if it's about lab, or ask me if you still have a question, and then tackle these last 2 chapters, because the tissues and the skin isn't really a very complicated, but again, once you get the lab completed for that, then you just need a couple days to really review that, and then you're pretty much good to go. And you could easily have this test taken. I mean, I believe it opens up on Monday morning. Try to have it done by like Wednesday, so you have a nice long weekend of really nothing to do. And that's still, if you started studying now, because today's Wednesday, is that you could spend the majority of Thursday, Friday evenings just working on that, still have and part of Saturday, and still have like nice Sunday and Monday off, and then study again on Tuesday, and just take it and get it done, and then kind of see where you're at. Right. So does anybody have any questions?
Yeah. Are we gonna have to identify any images in the test?
No, it's just like a written test. 50 questions. A lot like like the quizzes. I wouldn't say they are the quizzes by any means, but I mean, the quizzes are pretty easy questions. But I'd say most of them, are, if you can answer lab questions, you can lab and lecture, and you see how they're combined. There are a lot of structure function, like especially with chemistry, when I put those charts in there, like comparing protons, neutrons, and electrons to each other, I do it for a reason. You can review it, not review it, it's entirely up to you, but that's what I would focus on, is a lot of the material that overlap with lab. And that's the majority, I'm just telling you honestly, the majority of the questions. I'd say, it's a luck of the draw, because there are probably, I don't know, 125, 170 question pool, so it's kind of random as to what you're going to get, but they're mostly straightforward. There'll always be some that are a little more complicated, especially, things that I would prepare for are maybe describing bond types, and like a hydrogen bond especially, and what's unique about proteins. I mean, that seems to, kind of be a bugaboo with students, so don't let that stuff get too far away from you. The rest of the stuff that we just finished, like about cells, isn't really that hard. You make flashcards for all the different organelles, I mean, that's easy, and then the difference between diffusion and osmosis. I would make a flashcard, which are the processes, which are the passive processes, and then, when it comes to active process, what are the types of endocytosis versus phagocytosis? There's like, maybe in each of those chapters, are PowerPoints. There's maybe, like, especially with the cells, there's probably 5 or 7 PowerPoints that are kind of condensed and have some information that's a little physiologically something you have to kind of wrap your head around. And then that's really it. The rest of it is pretty much structure function things, organelle function. Know your phases of interphase of mitosis, and what cytokinesis is, and that mitosis really, is referring to division of the nucleus, which includes prophase, metaphase, anaphase, telophase, something like that. I wouldn't go totally crazy with extraordinary detail. Whatever's really reinforced in lab, then definitely when it's lab and lecture, you really need to know it, and that's where I would start. and then see how you do after the first exam. So good question. Anybody else?
Sorry, I spaced out for a second, but did you answer if the PowerPoints will have all the information for the tests?
Pretty much, yeah.
Okay, sorry, I had to look away
No,
from
I
that.
didn't say it before, but I think, like you're learning from the quizzes and things. Most of them are right from the notes. And that's why, I mean, sometimes every once in a while, I might ask a question that is kind of a little out there, but usually you can answer it based on the notes or lab. So, and if there isn't, then say, oh, we didn't cover that at all. And then you can let me know and maybe I'll throw the question out. But I will just say that on the most part, when people do that, I find where it's in there and they just overlooked it. So I might not sweat the small stuff because a couple points here and there, I gave you some extra credit opportunities later in the semester. So, and especially if you didn't do that well on that exam, that more than likely that's the one that's going to get dropped anyway. So, but I'll give you some chances later, for some extra credit, like completing the SSIs and things. But much more consistent with the notes that I think some of the eCampus, classes are. They pull some detail that is, I still never found it in their notes. I mean, I knew the answer, but I was like, where is it in the notes or in their video? And I never did find it. So I think we're much truer on this campus to asking you questions, about the notes that we provided, and or, how it's intertwined with lab. Occasionally there might be a question, but it's very few and far between, Levi. Right. Anybody else?
Okay,
Yes,
thank
when
you.
you say written test, is that more like short answer, fill in the blank, or is this
No, I just mean that this is a regular written test compared to the lab practicals where you're looking at models and yeah, I just mean it in contrast to that. So it's not, I don't use images, although that would be kind of fun to do, but no, I don't.
So are they multiple choice?
Just like, yeah, multiple choice. I think it says in the syllabus. Did you read the syllabus? Oh, snap. No, okay. It's multiple choice, true, false. I don't do a lot of matching. Occasionally I'd say more in, well, I would say that it very rarely do I ever have a fill in the blank, but and especially for you guys, I'm drifting over to AMP2, so my bad. In AMP2, sometimes there are a few fill in the blanks because we have some equations for things like blood pressure and cardiac output and some chemical equations, so the occasional, but not for you guys. I don't think there are any fill in the blanks. What else?
Are they
How
open
much
note?
you have? About an hour, I think, maybe 60 minutes with an extra 10 minutes, maybe 70 minutes for the exam. That's over a minute. It's essentially what you would have had if with the class met face to face. You would have had an hour and 15 minutes. Where'd you go, Levi? Did you hang up?
Oh no, I just wanted to ask, is the test open note?
No. Oh no. No, it says that all exams and things, and technically quizzes should be, but I'm not going to go, crazy with that. It should not be open notes. And, this is the thing. If you didn't study it, there's no way you're really going to be able to have time to look up all those questions anyways. I mean, that's why you have to study. And the subsequent exams will be all honor locked. And so, yeah, there are no notes.
I remember reading about that. I couldn't remember if that was this class or the lab though.
Yeah,
So thank you.
I think the lab, hey, Levi, who do you have for lab? And I'll be able to answer that, I think.
Dr. Walker.
Okay, I don't know. Her office is right next to mine when she's here occasionally. She might have changed her policy. Most of us don't require, don't allow any notes, in the class. And the reason why I don't is that even my lab exams are multiple choice, true, false, the same, format. With the eCampus ones, I will say, is, and I may also add is that she might be teaching a class for the Clearwater campus and not necessarily just for here because she's a part-time person and she kind of teaches, classes that are originating from different campuses, and sometimes they have their own kind of policy in their, natural sciences department as to how they want you to handle that. But the e-campus classes now that are totally strictly online, you use that Pearson book and, or you use that official lab book, they now let you use this 3x5 index card to write some notes on, but that's because their exams are 25, if not more, 20 questions with different parts, and you have to type in your answer. And that is consuming and more laborious, than just multiple choice true false, So because when we do these literally face to face down in the lab, it's all fill in the blank and you don't get to take any notes anybody. So we want to be kind of competitive with and fair compared to certain advantages they have for the online classes that have always been online and designed for being online forever, And then there's ours that are kind of modified from the COVID-19 to have formerly been face to face, and now we just moved it entirely online. So, because it's multiple choice and true and false and things, is that I generally don't allow you to have any notes, because that I think you should be able to do. If you had to do it all fill in the blank and write out stuff, that would be horrible. Then you'd have to give them some kind of notes that they could take with them. I think that would only be fair, quote unquote. So, good question. Anybody else?
How much time is given to take the test?
It says in there, I think you have 70 minutes, maybe 60 or 70 minutes, which is about one lecture period. When you hover over, it should, when you go to enter it, it says, you're time limited as to a certain amount. And I believe it's 70 minutes. If you want to give me a moment, I'll look. Hold on. I don't know if you can still see my page, if it's still sharing with my computer top here, but it might not be. So let me go to an exam because I'm pretty sure they're all the same.
It says for exam 1, we have an hour and 15 minutes.
75 minutes. Yeah.
Yes.
I knew it's somewhere around 70. All right. What else? I just can't impress, it's everybody's nature generally. They try to take as much time as they can to study, and then they take it on that last day. I'm just going to tell you that, I would try to do it, don't everybody wait till Sunday, because if something happens with our computer, it just becomes sometimes a nightmare. We don't have to worry about auto lock on this exam, but that's another issue, is they get flooded with, people accessing and requiring auto lock to, monitor, exams and things, and everybody seems to be having them scheduled and people taking them at the same time. And that's another reason why I spread it out, so that you wouldn't have, issues with that. I see some things in the chat, and I generally aren't able to do the chat at the same time as covering notes. So let me kind of quick go through there and see if there's anything. Not to complain, but I was just on a little bit of a sleep. It was, who did that? It was kind of hard to something. I don't know.
Oh, that was from the Kahoot quiz.
Oh,
You
to
talked about me
focus. Oh, that's
falling off.
okay.
Yeah.
Yeah, but you like jumped right back in. I have a quick question. Will we have to identify images in the test or quizzes? I don't know. Well, the quizzes you should know, because you should be taking quizzes now. Perhaps on a quiz, but I would say I don't think there's anything in exams. That's what Levi just asked. I know this question was from a little while ago. Do you recommend using your slides for most of the study? I do. And especially any of those charts and things that I create for you. I do that for a reason, and it generally, I find, works for most students. A&P is you learn it by repetition, by doing it over and over. If you don't go through these notes at least 4 or 5 times, you're really not committing it to memory. And, I know people are just like, okay, I'm just going to worry about exam 1. But, you have to, if you don't kind of deal with chemistry now, you're going to have to do the same thing over and over again because you never really learned it. You just memorized a few facts and got through it. And I'm telling you, you use chemistry. That's why we have to teach it, And I know it because I've sat in on nursing lectures and stuff too. So, thank you. Yeah, no, are her PowerPoints uploaded to the course? Oh my God, yes, they are. You haven't navigated through the course. They're all up there. The notes are up there and so are these Zooms. Yeah, understand what. Thank you for answering their question, Levi. Okay, thanks, blah, blah, blah. Thank you, off to work. Oh, she's working during my class. Thank you, your class, blah, blah, blah. It opens on my, yeah, they use the calendars, well, first of all, you have the schedule that tells you at least what it's due. But if you hover over the calendar, so, when you first sign in, you have, the heading on the top, you have course home, a calendar, I don't know, you have like grades and you have submissions and, different things up there, whatever, course content, right? So click on the calendar and put it in like the whole month mode, and that way you can see on there when you sign in on the course homepage over to the right-hand side, that should give you these little signals, that like, hey, this is due, this weekend or whatever. It tells you what's due in the next few days or the next week. So, there's nothing you can delete from there. The only thing you can do accidentally is enter into a quiz or an exam that might finally have been open because it won't open until Monday. And you accidentally go in and you start the clock, well, with the exam, you just got to then take the exam at that point, or if you only saw 2 questions, quick, close it out, let me know. I'll go in there, say, okay, you only looked at 2 questions or something, then I'll delete it and I'll let you restart it. But if you look at, a handful of questions, I can't even give you exact number on that. If I kind of get the feeling that you've actually looked at, a good number of those questions and then, you decided to exit and say, something happened with my computer, then, that's not really fair to the other students who went in and actually completed the exam in that time without being able to, come back into it again. So I'm just going to warn you, that's the only time where you can kind of boo-boo. If you do it on a quiz, you get 2 tries. And the same thing, if you entered into the first one, and you accidentally opened it and you didn't really look at anything except for like 2 questions or something, and you really weren't ready for it, just close it out and let me know immediately and I'll just delete it. I'll go in and look at it and delete it. But if fair is fair. Is the exam open to take now? No, it's not. It opens on Monday and it's open next Sunday until next Sunday. Oh, thank you. Someone said no, it opens Monday. Thank you, Rachel. I'm taking 3 classes and sometimes it does, it really, when, and that's, when the summer session is rough, it's condensed and it's a rapid 10 weeks, so you'll kind of sweat a couple LBs, because, it gets kind of hairy, and when you're taking multiple classes, you have all these different due dates. And I agree, hopefully your class gives you a good schedule. And that's what I would have on the wall, like with me in my my apartment, what am I saying? My office here, I have the wall and then I have me sitting at my computer, right? And, I sometimes, especially when I have a regular semester with multiple classes, I might have all the schedules for these classes like taped on my wall so I can quick review and you can check things off, put them in your phone with due dates. It is a lot to keep track of. It depends. Like ours, I tried to make it so everything is due on a Sunday night. So come the weekend, you should be like, what quizzes, or do I have a quiz or exam that's due this weekend? I don't have them due sometimes on a Friday and then sometimes on a Sunday. All your assignments will be due on Sunday nights. So that's a little less confusing. And again, you do have a schedule. So, have that printed out and then cross off your assignments as you complete them. That keeps you kind of tuned in every few days. And then I would do that with every class that I have. Is most of them hopefully have created a schedule for you that, again, the purpose is to print it out and put it by your workstation. So you can look at every week, when things are generally due on the weekend. So by Thursday, you're sitting your butt down and you're reviewing those things. and that's what I would do. To help keep yourself organized, because once those dates pass, it's, not everybody has a policy that you can go back in and maybe take a closed exam, like an exam one, and hey, but you can retake it, but you only, the highest you can get is an 80 or a 90. I don't even do that. You just need to take them. You need to just, at this point, when you're taking these sciences, this is preparation for these health sciences programs that are not flexible. So we need to really, prioritize things and, and get them done. And I would just tell you that generally, if you miss an exam, you try to make it up later. You're just robbing Peter to pay Paul, because you're trying to study new material and old material. And, it generally doesn't do well. So that's why I dropped one of the first 3 exams. So you can just keep moving forward. All right, anything else? Anybody else that does not have their cameras on that are hiding back there? If you have a question. George, nothing? You good? Caitlin? She's going, oh, no. All right. Well, if something comes up over the next few days, let me know. So Wednesday, I guess we will not Zoom. Just go through, download the Zooms if you want to redo the Kahoots and zippy to the point there. I think that would be way faster. You can do that over the next couple of days. And I'm just going to be, I'll be in and out online that evening, because I have other exams going on as well. So it's not like I'm very far away. And then we'll resume again on Monday. Enjoy your holiday. And I don't want to see anybody on the 6 o'clock news, myself included. So don't do anything I wouldn't do.
Bye. Thank you.
All right. You're welcome. Bye.
Bye. Thanks.
Thanks, Professor. Bye.